In the first part, what you see here is a side-by-side glimpse of two different worlds in the human body. A healthy state in which after you just enjoy your delicious meal, the food gets broken down into glucose and enters into the bloodstream, which signals pancreas to produce insulin in their specialized cell clusters called islets. Insulin then enters into the bloodstream, which acts like a key and regulates blood glucose level. Whereas in the second panel, type 1 diabetes is a lifelong condition in which insulin producing pancreatic beta cells are destroyed by cytokines, which are the chemical signals of body defense system. Because the defense system mistakenly attacks pancreatic islets, and there is no insulin production. This results in elevated blood glucose level in the body that leads to serious health complications. Type 1 diabetes is a chronic disorder which affects millions of individuals worldwide, including children and adolescents. And currently, there is no cure available besides insulin therapy, which itself cannot provide a complete cure. So, this condition demands an urgent need to develop novel therapies, which also includes identifying novel therapeutic targets. To address this problem, our lab recently identified Diras 2 as one of the novel proteins in the pancreatic islets, which was found to be activated due to cytokines that results in cell death. So what if removing Diras 2 would help survive pancreatic islets this autoimmune attack? To test this, we have two mice model in our lab. Wild type mice having a functional Diras 2 and knockout mice which lacks this protein. Pancreatic islets isolated from both of these groups were treated with cytokines for an overnight period in the presence of control condition. Pancreatic islets from wild-type mice having a T1D cytokines treatment were evaluated, whereas islets from Diras 2 knockout mice having a treatment of cytokines retained their shape, structure, and function. This was further quantified using two different cell death reporters, annexin and propedium iodide, which are used to measure early and late stage cell death in pancreatic islets. Surprisingly, a significant reduction of cell death can be seen in Diras 2 knockout mice compared to wild-type control. So these findings suggest that Diras 2 may promote cell death in pancreatic islets during stress conditions caused by these cytokines. And exploring a further role of Diras 2 would not only help identify novel therapies, but also enhance islet function. Moreover, if this Diras 2 contributes to cell death, then targeting this protein would stop or slow down the progression of type 1